DNA. Then when it goes to RNA, transcription takes place, and when RNA is converted to protein, translation takes place. And like protein is basically structural and functional biomolecule which is present inside the cell. And proteins are basically ribosomes. And when RNA gets converted to DNA, reverse transcription takes place, which is basically due to the viruses.
Correct. So my question is, let's take one or two convolute collectively test your concepts. So is it possible to make a DNA from a DNA under all circumstances, like normal circumstances?
No, sir.
Why?
So you say DNA gets reduced to RNA, so we can't like produce DNA from DNA itself.
We can't produce it, but it seems like we can, right? So you see this arrow, the process which we call replication. Can't we produce DNA from DNA? In fact, that is more common because every cell, when it divides, it has to produce more DNA and then segregate it among the daughter cells. Don't you think so? Yes, no?
Yes, sir.
So it's more common to produce DNA from DNA. And because DNA is more stable a molecule as we studied, it is a good way to store information, but it is not a good way to translate the information. So in between, there has to be a RNA that takes the message from DNA and then help the ribosome to produce protein. Basically, the information for this protein is present in our DNA. So any protein that is formed in our body, any enzyme, any factor, any receptor, anything, it has a gene in the DNA. So from DNA to RNA, it's transcription. And this process is reversible. You can make DNA back from RNA, but not normal cells can do that. Viruses, some retroviruses can do that. And they can do that through this special enzyme called reverse transcriptase. And the process is called reverse transcription. Okay?
Yes, sir.
Yeah. And from RNA to protein, it happens in our cell and it is called translation. So ribosomes help in this process. So ribosomes read the RNA and make the protein. Now my question is it possible to go back, like just like we can go back from RNA to DNA? Can we go back from protein and make RNA? Is that possible under any circumstance? Yes, no? What do you think, at least? Memona, what do you think? Can the information flow back from a protein to RNA?
No.
No, why? Why? What do you think? Why is it not possible? So it's not possible because proteins are formed by the ribosomes by reading the codons on RNA, correct? And there are more than one codon that code for one amino acid. So the code is degenerate for making proteins. So looking at a RNA, you can figure out what protein chain will be formed. But looking at the protein chain, you cannot tell what will be the RNA sequence from which it is formed. Do you understand that?
Yes.
So three nitrogenous bases. If we go to the process, we will go later on. But three nitrogenous bases are read to add one amino acid. Do you remember that translation process, people? Yes, no? So it's a bit heavy on concepts and facts, both chapters. So please be with me. If you find at any given time that you are not on the same page or you want to step back, take a deep breath and we can start again. Not a problem. So this is not a class where we have to cover a, a, a amount of syllabus. We are revising important concepts. So at any given point of time, we can step back and ask for some re um, discussion. Is it clear why we can never, the information cannot go from a protein to RNA? Yes, no?
Yes, sir.
Okay, cool. So similarly, uh, thank you so much, Mamuna, for explaining the central dogma. And then in this chapter, you know about DNA structure. So there are three components of every DNA molecule. A nitrogenous base, which can be of four types: adenine, thymine, guanine, or cytosine, and pentose sugar, which can be for RNA it is ribose sugar, and for DNA it is deoxyribose sugar, and a phosphate group. So if you see a phosphate group and the sugar, pentose sugar, make the backbone. The backbone keeps on going, and from this backbone, from the sugar towards the inside, protrude the nitrogenous bases. So they're like the stairs of a ladder, okay, stacking inside. So if you just talk about uh, sugar and a base without taking phosphate, then a sugar and a base is called a nucleoside because they protrude, the base protrude towards inside from the sugar. So it's called nucleoside. If you take phosphate also, so all these three form a single monomer of the DNA, which is called a nucleotide. So DNA is nothing but a long chain of nucleotides. So it's called a polynucleotide structure. Is that clear, everyone? Both of you on the same page with me?
Yes.
Yes, sir.
Okay, very good. And some important things about nucleus that I think these, so please keep your notes open about Franklin, who found the structure, what are the salient features. So would you like to quickly read the salient features? There are five, and we'll go read these four first. Ellen, would you like to read these salient features which are important for every DNA?
It is made up of two polynucleotide chains. Backbone is sugar phosphate bases project towards the inside. The two chains are anti-parallel to each other. The bases are paired to hydrogen bonding. Two chains are coiled in right-handed fashion. Plane of one base pair stacks over the other, double helix.
Correct. And you know about the pitch. This is called a pitch from one turn to the other. and one pitch of DNA has around 3.4 nanometer length of the DNA, and one pitch contains around 10 bases, 10 base pairs. So going by this calculation, you can calculate the distance between two base pairs, which is 0.34. That is the distance between two adjacent bases on the same strand. Is it clear? It
Yes,
is
sir.
not the distance between the bases on the opposite strands, it is the distance between the bases on the same strand. Okay. Then packaging, we know that it's supercoiled and this nucleosome structure is formed by DNA coiling over protein. Those proteins are called histone protein. There are core histones and there is a there is a side histone called H1 histone. These histone proteins are positively charged. DNA is negatively charged, so it can be bound on it and get more and more compacted, like it was shown here. So the compaction goes like nucleosome to chromatin fiber to a chromosome. Okay, clear? And based on this compaction of DNA, a DNA region can be can be called a euchromatin, which is a light stained or a loosely packed chromatin, and euchromatin is an active part of DNA which can get transcribed. Okay, so transcription can happen here. But in a heterochromatin, which is very, very densely packed, the DNA is not available to the molecular machinery that can read the DNA sequence and can transcribe a RNA. So that does not happen. So that's why they are called heterochromatized regions or inactive regions in terms of transcription. So euchromatin is transcriptionally active and heterochromatin is transcriptionally inactive. Correct? Then there were some important experiments that how this was found. So I would like to go one by one quickly. Just a quick summary of what was Griffith's transforming principle. Pamela, would you like to give a quick summary of Griffith's transforming principle? Are you there, Pamela?
Yes,
Yeah. Do you understand this or
yes, sir,
in
understood.
yeah? Would you like to quickly talk about it? Not in great detail, but just be quick.
Okay, so basically, like Frederick Griffith invented, I wouldn't say invented, but basically he named bacteria Streptococcus pneumonia, and it had like strain one and strain two, and then strain one was basically like smooth and strain, and this other one was rough and strain. And the strain one was basically like which produced smooth and shiny colonies on a central plate. It had lethal strain as well. And strain 2 was produced by rough colonies on a culture plate, which had non-lethal strain.
Okay. So, but what was this transforming principle? What happened? What was the experiment? So this is the experiment, right? What did he do and what did he saw?
So
Which
like
was interesting.
basically the S strain he injected in one of the mice, and the mice died. But when he injected the R strain in one of the mice lived. And so
I
like
think that
when
the S strain, the mice died, right? So you
yeah,
said the S strain where the mice died? Yeah. Okay, cool. Yeah, continue.
and when the estrine was heated, like when that was injected in the mice lived.
Yeah, yeah, correct, correct.
And then when S strain, which was heated, and the R strain, when that both were injected in the mice died.
So what does it tell you? Why is it interesting? Why should anyone bother? Alan, would you like to help
Yes,
me
sir.
out? Can you
Yes,
help
the
me out?
thoughts.
Why is this interesting?
So I didn't get you. What do you say?
Yeah, I'm saying that why should one bother? He put some S strain, then R strain, some mice died, some mice lived. Especially in this fourth experiment where he injected both of them together, the mice died. So what does it tell us? Why is it interesting?
Yes, sir. So it tells us that one strain takes up the infection and the other strain does not because of the polysaccharide coat surrounding it. The S strain does not take it up, but whereas the R strain takes it and then it transfers from the R strain to the S strain.
It transferred from the S strain to the R strain. Okay. So S
Okay,
strain
okay.
was very virulent. It was a strain that was producing polysaccharide, which you were talking about, and was producing the toxin, and that's why it was smooth. Our strain did not have that polysaccharide coat, so it was rough, but our strain is also not lethal. So when you heat kill the S strain, the mice should not die. And when you put R strain, the mice should not die. But when you put both of them together, the mice died. If you put both of them independent, one by one, the mice will not die. So something happened when you put them both together. Something from the S strain was taken up by the R strain that made the R strain virulent, right? And that something was not known at that time. And later on, every MacLeod and McCarthy, they did the biochemical characterization of Griffith's experiment, and they kind of said that it is DNA that is the genetic material. But their conclusion was not taken very seriously, so that's why Hershey and Chase experiment was the was taken as the unequivocal proof that DNA is the genetic material. And so, Alan, would you like to tell about Hershey and Chase quickly in 2 3 minutes? What did they do?
So to find out where what is the genetic carrier, if it's a protein, if it's a DNA, or um, if it's an unlabeled protein code, they added radioactive material to both the bacteriophages, and it was found out that only the DNA carried out the radioactive material, whereas the protein did not. So they concluded that DNA was a hereditary carrier material.
Yeah, so they saw that when they labeled DNA radioactively so they used, what did they use to label DNA?
Where is radioactive sulfur?
Phosphorus.
Phosphorus, yes.
DNA has phosphorus and protein has sulfur. And DNA does not have sulfur and protein does not have phosphorus. That way you can exclusively label either the DNA or the protein. Okay. And then they realized that the signal of radioactivity comes from the bacteria when you label it with phosphorus, and it does not come from the bacteria when you label it with sulfur, which means protein does not enter the bacteria, but phosphorus does, which means DNA enters the bacteria, and hence DNA is the genetic material of the virus that enters the bacteria. Make sense? And hence the genetic material was figured out. And these were some properties of genetic material, which is it should be self-replicating, it should be chemically and structurally stable, it should also be flexible to changes like through mutation, which is required for evolution, and it should be able to express itself in the form of Mendelian characters. Okay, cool. So till here, let's go for some questions. Okay, what's it? So let me share the screen. Okay, molecular basis of inheritance we are doing. Can you all see the screen that I'm sharing
No,
now?
sir. Yes.
No?
Yeah, now we can.
Now you can see. Okay. Cool. So we'll go through the same thing that we did yesterday. We'll read some, so I've chosen some questions, I've marked some important questions which are confusing, or we'll go in an increasing order of um, difficulty. And I'll first, don't try to take more than 90 seconds, is what is the point. Do it fast and try to do it correct. Okay. Are you ready, everyone?
Yes.
So the first question is kind of a very simple. Let's start with the first question. I think both of you will get it right. I'll read it for you. The final proof of DNA's genetic material came from the experiments of whom? A, B, C, or D? Quickly in the chat, just to test as a tester if this thing is working. Question number one, quickly. What's taking? We just discussed it right now. Who gave the final proof? Are people not typing or I'm not receiving? Am I audible, people?
Yes, sir.
Yeah, the first question. Is it that difficult?
So I sent the answer.
I have not received anything. Why is that happening? Have you sent it to me?
Yes, sir, that's my
Yeah.
mistake. I sent it to the.
Okay. So now I received. Yes, that's correct. Mamona, what about you? We are just testing by this that you people are sending it correctly to me.
Option D.
No, Griffith told about a transforming principle. He could not tell whether that transformation was happening because of a protein, a DNA, a RNA, or what. So it came from Hershey and Chase. Remember? Just now we talked about the bacteriophage experiment where they labeled DNA with phosphorus, labeled the protein with sulfur. Correct? Correct?
Yes.
Yeah. So it is option A. Okay. Good. So now let's start. Question 2. Again, simple but a little confusing. So Mamuna, would you like to read question 2? And when Mamuna will finish reading, you both will have 90 seconds to give your answers. Yes, Mamuna. Over to you.
If there are 999 bases in RNA that code for a protein with 333 amino acids and the base at position 901 is deleted such that length of RNA becomes 998 bases, how many codons will be altered? Option A 11, option B 33, option C 333, option D 1.
Yes. Okay, very good, Mamuna. I received an answer. Correct. Both of you, correct answer. The answer is option B 33. And Mm, I don't think this requires explanation. It's simple that for every amino acid there are three bases which are read, right? As they say, 999 bases give 33 amino acids. And if there is a base at position 901 which is deleted, okay, so now from that 901 onwards there will be alteration. So from here onwards there are 99 bases that will be altered. So 33 amino acids will be altered, or 33 codons will be altered. Correct? Both of you thought the same, correct?
Yes, sir.
Perfect. Now let me take a slightly confusing question. Yes, question number 10. Would you like to read it? Sorry, Alan?
A molecule that can act as a genetic material must fulfill the traits given below, except it should be able to express itself in the form of Mendelian characters. It should be able to generate its replica. It should be unstably structural and chemically. It should provide a scope for slow changes as required by evolution.
Yes, answers. Yes, Alan just changed the answer, but the second answer is correct, Alan. Remember, I have not received your answer. Yes, perfect. So the right answer is C. It should be, the question is asking except. So what all should be the features of a genetic material, or what all should the genetic material fulfill except? So just by adding except, they are asking you which is not a characteristic. So read the question carefully, okay? But C, it should be unstable. It should not be unstable, either structurally or chemically. Okay, is that clear?
Yes.
Yeah, great. Now let's go to some important uh, concept-based questions. So this is question number 20, but the way you have to read it, can you see question 20? So what it says is that in sea urchin DNA, I will read that, which is double-stranded, 17% of bases are shown to be cytosine. So just remember here that 17% is cytosine. You can write down in your notebooks right now. 17% of bases are cytosine. And then the percentage of other three bases will be what? Quickly. A, B, C, or D. Just look at the options and tell me. If 17% are cytosine, 1 7. Yes, Ellen, correct answer. And good that it's passed also. Very good. Mamana, we're waiting for your answer. Please do not hesitate in letting me know if you find any difficulty with any question. If you can discuss the concept for that question, not at all a problem. Are you finding it difficult, or are you confused or stuck somewhere? We can discuss that. Am I audible, Mamon? I think there's some issue with Mamon not being able to hear me. Okay, so Ellen, why not you tell us what is the concept behind solving this?
So G and C are paired by double hydrogen bonding, and A and T are paired by triple hydrogen bonding. So G will be equal to A, which is 70%, and then you subtract 17
G
into
will be
2.
equal to C, not A.
Sorry?
G
I,
will be equal to C.
yeah, I mean, sorry, G equals C. So then if you subtract 100 from 34, you get 66, divide that by 2, and you get 33.
Yes, perfect. So that's a so basically the total bonding will be between AT and GC, which is 100%. Okay, so 100% only these four bases can be present. So and C is only given, so you know that G will be as much as C is, and then GC together subtracted from 100 as Alan is saying correctly, and then divide by 2. Perfect. Good. Okay. So followed by let's read 21 as well. It's an important question, has come multiple times. Even in boards, many questions are asked on operons and transcription translation. Yeah, so Ellen, would you like to, okay, let Mamona rejoin again. She is joining. She's in that waiting area. Yes, Mamona has rejoined. So are you able to hear us, Mamona? Mamona, am I audible to you? Because you are not audible to me. I don't know about Ellen. Ellen, is Mamona audible to you? I see her
No, sir.
hand. Yeah. Okay. So, Mamina, if you can hear me, you are not able to speak, but you can hear me, you can write your answers in chat, it will work. So please write in the chat if you can hear me. Okay. I don't know. Anyways, okay. So Ellen, would you like to read question 21?
Which one of the
Mamina?
following is wrongly matched? Transcription, writing information from DNA to tRNA. Translation, using information in mRNA to make protein. Repressor protein binds to the operator to stop enzyme synthesis. Operon, structural genes, operator, and promoter.
Yeah. Which one do you think was incorrectly matched?
So option A.
So Mamina,
Okay.
yeah, just write in the chat because she's there. Mamina, we cannot hear you. If you can write your answers in the chat. So there is a reason I chose this question because you might. Can you read this question? Do read this question again, Ellen. So you're not incorrect. You're correct, but do read it again and see if I'm not receiving Mamuna's answers. So I don't know if she can hear me or not. But yes, A is the correct option, which is A is wrongly matched. But will you also look at option D? Do you think D is correct? It is correctly matched.
So I think it's right, right? I'm not sure, but
The sequence correct?
the sequence. No, yeah, it's right, right? The D option.
D is incorrect. Okay, so they're asking which is wrongly matched. So operon is wrongly matched when you say structural genes, operator, and promoter. Okay, it is operator, promoter, and structural genes. It
So
is
then,
not
A,
structural.
so then A is also wrong, right? Because it's supposed to be mRNA.
No, no. A and D both are wrongly matched. For
But we
question
can't.
21,
Yeah, but we
yeah,
can't
you
choose.
cannot pick. Yeah, you cannot choose both. But when this question came, and in the year 2014, anyone who selected option A or D both got marks. Okay, you didn't because it says that you can only choose one, but just know that option D is also wrongly matched because the sequence is not correct. Okay,
Okay, sir.
yeah, but you're right. You went for the most, like, which is more wrong, and that is A, obviously. So if you would have chosen A in your exam, you would have got marks. This question would have been cancelled because even D is not the correct one. That's why I chose this question. Just tell you that it might happen sometimes in the entrance, so just be aware. Okay.
Yes, sir.
Yeah. Cool. Now I'll go to some difficult conceptual questions, and yeah, it is question number 34. A simple one, but just read. So I think Mamuna is not able to hear me, Helen. So you have to read questions. We can do alternate. One you will read and one I
If one strand of DNA has nitrogenous base sequence as AT-CTG, what would be complementary RNA strand sequence?
will. Okay, please
TTAGU?
don't read that. And please
Okay,
write the answer in the chat, okay?
yes,
Because
sir.
maybe Mamuna join us in answering it. Yes, that's correct. That's perfect. Right answer. It's option B, UAGAC. Okay. And I think it's simple because in RNA instead of T there is U. Correct. I think that was your logic as well.
Yes, sir.
Got it. Now let's increase a little bit.
Sir,
Yes, Mamuna, good to hear you. Go back.
sir, can I leave the meeting because I have exam tomorrow?
Yeah, sure. It's up to you. So basically, after doing some questions, we'll be going back to the chapter, the notes, and we will take evolution, discuss something, and come back and do some questions. So that's that we will do.
Okay, sir.
Yeah. Okay. Cool.
Thank you, sir.
Okay. So Ellen, you're fine with discussing, right?
Yes, sir.
Yeah. Okay. So let's. Oh, so sorry. I lost it again. Give me 1 second.
Okay, sir.
Um, okay. Can you see the screen that I'm sharing?
Yes, sir.
Yeah. Okay. Um, cool. So, one moment. Okay, 41. Question 41, Alan. It has one concept in it. So you can read and tell me the answer.
So 41, right?
Yes, 41.
Which one of the following palindromic base sequence in DNA can be easily cut at about the middle by particle restriction enzyme?
Yeah, so since all are not visible to you, two options are on the other page. So what I'm doing is,
Okay.
let me see if I can accommodate all in the same page. You can just see it. Can you see these other two options here?
Yes, I can.
Yeah. So A, B, C, and D. So the question is, let me also see if I can accommodate the question. Can you also see the question? Can you read it?
Yes, sir. That's option C, right?
Yeah. So which one do you think can be cut by a particular restriction enzyme?
Yes, I
Yeah,
think it's
come
option
on.
C, 5GATC.
Yes, it is option C. And if I ask you, going one step further, which two bases do you think it will cut between more?
So I think between A,
A, A, to T.
no,
But the cut will be, yeah. It cannot be between AT because AT is at the center. That's why I asked. Remember when we discussed, I said that restriction enzymes do not cut exactly at the center. So they cut away from the center. So either it could be AA or between TT.
okay, sir.
You understand?
Yes, sir.
You have to go away from the center and then cut between the same bases on both the strands. That is the idea. And why is that? Because that kind of cutting will leave what?
Complementary base strands.
Oh, it will give rise to flanking regions which are called
So I didn't get you what?
sticky ends. Sticky ends.
Yes, sticky and unpaired hydrogen bonds.
Yes. So that can only be possible if you are cutting away from the center, either to the left or the right, but between the same bases on both the strands. Make sense?
Yes, sir.
Yeah. Okay, great. So coming back to the next question I chose was, yes, question number 64. If you can read question number 64.
Protein synthesis in an animal cell occurs only on ribosomes present inside the cell, only on ribosome attached to the nuclear envelope and endoplasmic reticulum, on ribosome present in nucleus as well as cytoplasm, on ribosome present in cytoplasm as well as in mitochondria.
Mhm.
Yes, just a second.
Check.
So I think it's option. So I said option. I think it's either option B or C. I'm not sure about this one.
B or C.
No, no, no. I
Okay.
think it's either, no. C or D. I'm not sure which one exactly.
C or D.
Sorry, sir.
Yeah. So you're saying you are not
Yes,
sure
sir.
between C
Yes,
and
sir.
D.
I'm not sure on this one.
Yeah. So let's discuss this a bit, okay, so that it stays as a concept in your head. So protein synthesis, they are asking. So what are the, where does no, protein synthesis happens from mRNA, right?
Yes, sir.
Yeah. And what we have studied, that process is called translation, and that happens on ribosome But ribosome, is it present in the nucleolus?
No, it's present in the cytoplasm, so it's option D.
It's present in the cytosol. So C is a C is a just twisted option. So it says that ribosome
Yes, sir, the new,
present in nucleus. That's wrong. So C option cannot be the option. What do you think is the right answer?
D, because it's the
D.
mitochondria and cytoplasm.
Yeah. So ribosomes present in cytoplasm as well as in mitochondria. So mitochondria also synthesizes some proteins because it has its own DNA. Correct?
Yes, sir.
Yeah. Good. So its own DNA gets transcribed into RNA, and then that RNA produces protein. So do you understand
Yes, sir.
where the confusion was in this? It was a little confusing, but good. You did it well. Let's see some more like this. These are all simple fact-based. We are not going for many fact-based questions because that I think depends on how good you read the NCRT. So please read the NCRT. I will
Yes,
go
sir.
for yeah. I will go for yes. Question number 99. If you remember the pneumococcus experiment,
Was this there in the textbook?
sorry.
No, I don't remember reading this in the textbook though.
Okay, so let me first give you. So I said that you have to read the textbook. Are you asking what I said about textbooks?
No, sir, I don't remember seeing this question, like this exact
Oh,
wording in
cool. So
textbook.
we'll come to it. We'll come to it in a bit. So let's do yes. Question number 105. It is concept, but also you have to remember things. Question 105. Can you see and read question 105, Anna?
Yes, sir. So the eukaryotic genome differs from the prokaryotic genome because the DNA is complex with histone in prokaryotes. The DNA is circular and single-stranded in prokaryotes. Uh, Repetitive sequence of present eukaryotes, genes in the former case are originated into operons.
What do you think?
I think it's option B, right? The DNA is circular and single-stranded in prokaryotes.
Yes, perfect. That's option B. So if you see, option A is wrong because there is no histone in prokaryotes that complexed the DNA.
It's a lack of
Option
nucleus and
option C is actually correct. Repetitive sequences are present in eukaryotes, but that is not the reason why it is different.
yes, sir.
Because these repetitive sequences might also be present in prokaryotes.
Prokaryotes as well. It's bad in both.
Yes. D is so C is correct in option, but not the reason. Correct reason. So D is genes in the former case, which is eukaryote, are organized into operons. No, genes are organized into operons in prokaryotes. Is it clear?
Yes, sir.
Yeah, correct. So that's why it's B. Perfect. And then you have yes, read question 120, 120.
So which one? 120.
120, 120.
If the DNA coordinates are ATG, 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 and a cytosine base is inserted at the beginning, then which of the following will result? CAT, GAT, GATG. A nonsense mutation C, ATG, 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 CA, TGA, TGA, and TG.
So it is a concept of how it is read. So tell me. So read the question again and tell me the answer. Now read it for yourself, not out loud.
So I said option A.
Yes, correct. It's option A. And why do you think so? Because whenever you add something anywhere, You still read the codon 3, right? The first
Yes,
thing.
sir.
So cytosine is inserted in the beginning. So now the frame will shift from ATG to CAT. The next will be GAT, and the next will be GAT. So just to incorporate that, so that in fact option A is also not completely correct. Can you tell me why?
Should be like tenable complementary base mass as well.
No, because the last one has 4 GATG. It should be CAT, GAT, GAT, and G should be separate, right?
Oh,
Because
okay,
it is read
that
3
one,
at a
okay.
time. Yeah, but that is the
Yes,
most
sir.
correct. The others are even like they're not
Yes,
correct at all.
it's not.
Yeah, do you understand why it is not the 100% correct option?
Yes, sir, because D and G should be separate in option A.
Yeah. Yes, correct. Right. Right. So then there's more fundamental. One more question we will take before. Mm, yes, 132. It is a factual question, but it also is asked in various other ways. 132.
During DNA replication, the strands separate by DNA polymerase, topoisomerase, unbindase or helicase or gyrase.
So basically, it's
Uh,
asking what separates the DNA during
so which,
replication.
so I think it's pretty much DNA polymerase.
DNA polymerase replicates the DNA. That is what people, because DNA polymerase is talked about the most in NCRT. So to make this mistake, that's why I thought that I should include it. DNA polymerase, as the name says, it polymerizes the DNA. So it makes new DNA. Okay. So it is helicase
Oh,
because
okay,
the
okay.
DNA is helical, right? It's double helical.
Yes, sir.
So now, so this is an old question, but now I've also seen this question in recent exams, but they don't give you the option unwind days because unwind days is not a official name of that enzyme. It is called helicase. It is called helicase because it opens the double helix. Okay.
Yes, sir.
And
Yes,
when
sir.
it opens the double helix, it separates the two strands of DNA. Make sense?
Yes, sir.
Hmm. Good. Um, and just one last question. Yes. Question number 141. 141.
In
Clear?
genetic,
Anything?
yes, sir. In the genetic dictionary, there are 64 codons as 64 amino acids are to be coded. 62 types of RNA are present. There are 44 nonsense codes and 20 sense codes. Genetic code is triplet. Sorry, Princess option D.
Option
D.
D. Okay. So what is the problem with A? You're correct. Right answer is option D. But option D is not the most optimum option. But A, B, C are very, very absurd and wrong to many levels. So A is wrong because there is not 64 amino acids. There are only 20 amino acids that we know of. Correct?
Yes, sir.
What is wrong with 60? What is wrong with option D?
Option which, sir? B.
Yeah.
Sorry, I just said option D because I remember seeing this word in that textbook, genetic code being triplet.
Yeah. So you also should know because maybe option D will not be there, but for the same question you can have more options, right? It can be correct. So there are 20. The wrong with A is that there are 20 amino acids to be coded. In B, there are not 64 types of tRNA. There are only 20 types of tRNA because for every amino acid there is one type of tRNA, right?
Yes, sir.
Which transfers it. With option C, what is wrong? There are 44 nonsense codons and 20 sense codons. No, there is no concept of nonsense and sense codons. So C is wrong. So the only option which is right is that genetic code is triplet. Whereas it has nothing to do with 64 codons. If codons were, let's say, 84 also, still like genetic code has to be triplet only, right? So it is not a very connected option, but it is the only correct option. Do you understand,
Yes, sir.
Alan? Yeah,
Yes, sir.
cool. Fine, great. So you're good to go with this chapter. Your concepts are clear. So well done. And please keep revising. And what I will do is quickly, we also will take some evolution questions. So in genetics, evolution is a hot topic for entrances and boards both, though its weightage is lesser than chapter 5 and 6 from this unit, but let's not leave it. So what I will do is, let me share the notebook again. Can you see the notebook?
Yes, sir.
Yeah. And let me go to evolution. And if you have any, do you have notes with you? The notes for evolution that we did in class?
No, sir. I joined after this class.
Or you joined after this? Okay, cool. So, do you have NCRT with you?
Yes, sir.
Can you open NCRT chapter 7 and go through just the chapters, like just go through the headings and the concept, because you must have done this chapter. And if you find any topic which is specifically of difficulty to you, then we can discuss them first. Okay. So I give you 5 minutes, okay, to go through the chapter by your own, and I'll be available for the next 5 minutes. I'll be here only. So kind of a self-study session.
Yes,
So
sir.
for
Okay.
5 minutes, go through the book and note down the topics which you find are difficult. Okay.
Okay, sir.
Yeah. And your time starts now. Then we can discuss some of those topics, and then we can take some questions. Even if we cannot take the questions today, and we just end up discussing your doubts and difficult topics, we'll take it in the next class. Not a problem. Okay, Alan?
Yes, sir.
Yeah, so go over it.
Yes, I'm done.
Okay, great. So do you find any specific particular topic that is difficult?
Yes, I can just talk about the Hardy-Weinberg principle.
Hardy-Weinberg principle, right?
Yes, sir.
Okay, cool. Let me see if we have talked about it in this one or the next. Divergent natural selection. Oh, okay, so it's in the next, I guess. Okay, so Hardy-Weinberg, right? Yes. Yeah. Can you see the screen?
Yes, sir.
So first, before we discuss about Hardy-Weinberg principle, let me also ask you if you know about what is allele frequency. You know what are alleles, first of all?
Yes, sir.
Yeah. So alleles basically are what are
They are paired characteristics present in the gene that represent themselves in genotypic or phenotypes.
yes, perfect. So they are paired characteristics, and in other words, if we put very simply, I can say that are uh, alleles are slightly different forms or a very varied form of the same gene. Okay. So for example, when we do Mendel's crossing, we say that capital T is an allele for tall character and small t is an allele for short character. But both capital T and small t are genes which talks about the height or which determine the height of the plant, correct? So the gene is same. These two alleles are slightly different in their sequences. So one will make protein which will lead to tall height, and other will make protein which will or will not make that protein or will make some other protein. Is it clear?
Yes, sir.
So that is allele. Now we are talking about that for every gene we know or we assume. So for this Hardy-Weinberg principle, we are assuming that for every gene there is at least two alleles present, right, in pairs. Yes or no? Correct?
Yes, sir.
Perfect. So two alleles are present, one allele on each chromosome. So for every chromosome there are like one pair, two chromosomes, one coming from the father, one coming from the mother. And one allele for the same gene will be coming from the mother, and one allele for the same gene will be coming from the father. So there are two alleles for every gene in every organism. Is that clear?
Yes, sir. So the frequency would be I think it'll talk about the representation of the alleles in a gene.
Yes. So the Hardy-Weinberg principle says that in a population, no matter how big, so the bigger the population, the better it is. But no matter how many genes we talk about, in a population, there is always a genetic equilibrium that will reach. So it says that allele frequencies in a population are always stable and remains constant from generation to generation. Okay. So it simply means, in other words, that the total genes and their alleles in a population remains constant. For example, let's talk about the eye color in humans. So there are now around 8 billion people on the planet, right? 8 billion humans are there. But you think that there are 8 billion different eye color characteristics?
No, sir.
No, right? If it comes to hair texture or hair color or skin texture and skin color, do you think that they are all as diverse as the number of individuals in a population?
No, no,
No,
sir.
right? There are always fixed number of characters. For example, even if it's something as trivial as the shape of nose or shape of the chin or shape of the ear, there cannot be infinite ways. So it's limited. Characters are limited by alleles and genes. So you will see what is different, what makes, but still you will find every human will be unique in its own way than the other human being. Correct?
Yes, sir.
Except for identical twins, which are very, very similar. But you will also see people who who do not belong to the same family, are not related to each other, um, live in completely different regions on the planet, but still may end up looking similar. Correct?
Yes, sir.
It happens. That's how the doppelgangers or the body doubles of these celebrities come up, right? They look very,
Yes, sir.
very similar
Yes, sir.
to
Yes, sir.
some famous people. But they will also have differences and there will be unique features that they hide in order to look like their celebrity or the role plays. Okay. So this happens because the frequency or the total gene pool is constant. Now, what it talks about is, let's say in a diploid organism, that sum total of all allelic frequencies is 1. This is what this Hardy-Weinberg principle says, that mathematically, if you do the sum of all the allelic frequencies for any given gene, that will be 1. Okay. Now, in diploid organisms, we know that there are 2 copies of every gene, which means 2 alleles. So taking an example of capital T small t. So if it is, so let's say, and Hardy-Weinberg principle says that there is a formula by which you can find out the allelic frequency for any allele. So let me first say that capital T is denoted by P here, okay? P is the frequency of capital T. Is it clear, Alan?
Yes, sir.
And Q is the frequency of small t. Now P and Q must be some numbers, but here we are just defining it as some constant alphabet. Okay, we don't know what number is it. So mathematically, P denotes frequency of allele capital T and Q denotes the frequency of allele small t. Make sense?
Yes, sir.
Now let's come to what is possible in biology. So in biology, either an organism can have capital T, right? In that case, the frequency becomes P into P. So P is the frequency of capital T and P is the frequency of capital T again. So P into P, which is P square. So if it is a homozygous dominant capital T, the frequency becomes P square.
Yes, sir.
Alan, same, the chances of both small t coming together as frequencies, the frequency will be Q square, right? Q into Q, correct?
Yes, sir.
The third condition could be a heterozygous form, where one capital T is there and one small t is there. But it can happen in 2 ways. One is that mother is giving small t and father is giving capital T, or mother is giving capital T and father is giving small t. So both these ways biologically are the same thing. If we see the biology, that remains the same. That does not matter. But mathematically, there are 2 different scenarios, right?
Yes, sir.
So if it is small t, capital T or capital T and small t, for t it will be P and for small t it will be Q. So the frequency is P into Q, but it can happen 2 times. So 2 into PQ. Is it clear? So 2PQ. So for a heterozygous condition, the frequency becomes 2PQ. Now, where does this 2 comes from? Just to explain this, I was explaining that mathematically there can be 2 ways by which heterozygous alleles can come together. But biologically, both these ways are not any different. They are the same. But since we are defining a mathematical equation here, we have to take that into consideration. Correct, Alan?
Yes, sir.
Okay. So taking all of these 3 frequencies, for homozygous dominant, it is PQ. For homozygous recessive, it is Q square. Sorry, P square, Q square. And for heterozygous, it is 2PQ. Now, as the definition says, sum total of all allelic frequencies is 1. Then we write the equation P square plus 2PQ plus Q square is equal to 1. And you have already studied in your mathematics, this resembles a formula. A square plus 2ab plus b square, and it is equal to a plus b whole square, right?
Yes, sir.
So this is how we can mathematically find the allelic frequency of any given allele in a population. Because in a population, the chances are three: homozygous dominant, homozygous dominant, homozygous recessive, or heterozygous. Correct? So you understand where this formula came from?
Yes, sir, I understand. By combining all of these, p square, q square, and 2pq.
Yeah, correct. Now, would you like to do a few questions on this? Yeah.
Yes, sir.
Let me find a question and I'll stop sharing this and we will quickly take a question. Yes. So, okay. Can you see the screen that I am sharing, Helen?
No, no, sir. Yeah, now I can see. Yes, sir.
Yeah. Okay. Cool. Great. So, can you read question number 52 and see if you can solve it using the same?
At a particular locus, frequency of A allele is 0.6 and that of small a is 0.4. What will be frequency of heterozygous in a random mating population at equilibrium?
Yep. Can you solve this?
Yes, sir.
Yeah.
So the option D is 0.48.
Option
D is
D.
0.48. D, yeah.
Perfect. So I think you got it right.
Yes, I squared 0.6, 0.4 and subtracted from 1.
Perfect. So great. So you understand how to use that formula
Yes, sir.
to find any allele. So these kind of questions will come on Hardy-Weinberg equilibrium. Okay.
Okay, sir.
Okay. Cool. So let me see if there is one more. A shortlist with one more question related to this. Hmm. Old question on Hardy-Weinberg. Yeah. Okay, we'll do some more questions on Hardy-Weinberg and others, but there's one more question which I would like to see from human evolution. It was from yes, question number 55. Can you think what is so? It is a question that the answer is not directly in the NCERT, but you have to think. It is more logical. Can you read this? Question number 55.
What kind of evidence suggests that man is more closely related to chimpanzee than with other homoid apes? Evidence from DNA extract from sex chromosomes only, comparison of chromosomes morphology only, evidence from fossil remains and the fossil mitochondrial DNA alone, evidence from DNA extract from sex chromosomes, autosomes.
Yeah. What do you think could be an answer?
So option A.
So option A says that evidence from DNA extracted from sex chromosomes only. So sex chromosome is just X and Y. So it could not be the correct answer because most of our genes that make us similar to chimpanzee are present on autosomes, also on sex chromosome. But B is wrong. You cannot comparison of chromosome morphology only. C is also wrong. Evidence of fossil remains and fossil mitochondrial DNA alone. No, because humans and chimpanzees both are alive on the planet. So we don't have to look at fossil remains. So option D is the most correct option, where we look at the DNA extracted from sex chromosomes and autosomes all. So if you look at all the chromosomes that humans have and chimpanzees have, you will realize that our genes are, our DNA is 99.9% similar. Our genes are similar to a chimpanzee genes. So it is just the 0.1% that makes the difference. Okay. And recently I was going through some papers and a very interesting picture of a chimpanzee. The chimpanzee, if you see their hands, they're all black. They are very pigmented. So their fingers are black. Their knuckles are black. So a chimpanzee got this rare disorder where it lost all the pigments in its fingers. And to your amazement, you will realize that the moment a chimpanzee loses pigmentation in the fingers look exactly like a human finger, like our the color of our fingers is. Okay, do you understand that?
Yes, sir.
So that is again one very closeness which we can reveal through disorders. Okay, so that's all we'll do today, and we'll continue from here in the next class. So Ellen, for the next class, because you have time this week, let's also include biotechnology.